Imagine our DNA as a vast library. It holds the instructions for life, written in the language of genes. Each gene is like a recipe, telling the cell how to make a specific protein. Proteins are the workhorses of our cells, carrying out a myriad of functions. The process of using DNA to create proteins is called gene expression. First, DNA is transcribed into mRNA. In the cytoplasm, ribosomes read the mRNA and assemble amino acids into a polypeptide chain. This chain folds into a functional protein. From DNA to RNA to protein, this flow of genetic information is central to all life. The journey from DNA to protein is remarkable, but the story doesn't end there. Proteins often need further adjustments to function optimally. This is where post-translational modifications, or PTMs, come in.
PTMs are like fine-tuning a machine after it's built. They involve attaching small chemical groups to specific amino acids, altering a protein's activity, stability, and interactions.
Think of it like this: a car fresh off the assembly line can be modified for different purposes. Similarly, PTMs provide proteins with specialized functions.
PTMs are essential for virtually every cellular process, from cell signaling to DNA replication and repair.
Imagine a light switch that can instantly turn on or off a light bulb. That's phosphorylation in the world of proteins. It's one of the most common and important PTMs, acting like a molecular switch.
Phosphorylation involves the addition of a phosphate group, PO4, to specific amino acids, usually serine, threonine, or tyrosine. This addition can change the protein's shape, charge, and activity. Think of it as attaching a small charged post-it note to the protein, altering its properties.
The addition and removal of phosphate groups are tightly controlled by enzymes called kinases and phosphatases, respectively. Kinases add phosphate groups, effectively turning on the protein, while phosphatases remove them, turning off the protein.
Phosphorylation is crucial for a vast array of cellular processes, including cell signaling, metabolism, and cell cycle regulation. For example, the hormone insulin, which regulates blood sugar levels, exerts its effects through a cascade of phosphorylation events. Dysregulation of phosphorylation is implicated in many diseases, including cancer and diabetes.
Imagine a cake adorned with intricate frosting and decorations. That's glycosylation in the protein world. It involves the attachment of sugar molecules, or carbohydrates, to specific amino acids, creating a diverse array of sugar coatings on proteins.
These sugar coatings, called glycans, can be simple or complex, influencing a protein's folding, stability, and interactions with other molecules. Think of them as adding a sweet and sticky layer that can either protect the protein or help it stick to other molecules.
Glycosylation is particularly important for proteins secreted from cells, such as antibodies and hormones. For example, the glycosylation pattern of antibodies influences their ability to recognize and neutralize pathogens. In fact, many therapeutic proteins like erythropoietin, which stimulates red blood cell production, are glycosylated to improve their efficacy.
Glycosylation is a complex process, with various types of sugars and attachment sites. Aberrant glycosylation is linked to many diseases, including cancer, inflammation, and neurodegenerative disorders. Understanding the intricacies of glycosylation is crucial for developing targeted therapies.
Imagine a recycling symbol on a plastic bottle, marking it for degradation and reuse. That's ubiquitination in the cellular world. It's a fascinating process that tags proteins for destruction.
Ubiquitination involves the attachment of a small protein called ubiquitin to a target protein. This tagging is carried out by a sophisticated enzymatic machinery, ensuring that only specific proteins are marked for degradation. Think of it as attaching a molecular death warrant to the protein.
Ubiquitin-tagged proteins are then recognized and degraded by a cellular machine called the proteasome. The proteasome acts like a garbage disposal, breaking down the tagged proteins into their constituent amino acids, which can then be recycled to build new proteins.
Ubiquitination is crucial for regulating protein levels within cells, ensuring that damaged or misfolded proteins are removed and controlling the progression of the cell cycle. Dysregulation of ubiquitination is implicated in many diseases, including cancer and neurodegenerative disorders, highlighting its importance in maintaining cellular health.
Imagine a dimmer switch that can fine-tune the brightness of a light bulb. That's acetylation in the realm of gene expression. It's a reversible modification that primarily targets histones, the proteins around which DNA is wrapped.
Acetylation involves the addition of an acetyl group, or COACH3, to lysine amino acids. When histones are acetylated, the chromatin structure, the packaging of DNA and histones, becomes more relaxed. This relaxation allows easier access for transcription factors, the proteins that control gene expression.
Think of it like this. Acetylation loosens the grip of histones on DNA, making the DNA more accessible for gene expression. Deacetylation, the removal of acetyl groups, has the opposite effect, tightening the chromatin and repressing gene expression.
Acetylation plays a crucial role in regulating gene expression, influencing a wide range of cellular processes, including development, differentiation, and disease. Dysregulation of acetylation is implicated in cancer and other diseases, highlighting its importance in maintaining cellular balance.
Imagine a single dot of ink on a page, subtly altering the meaning of a word. That's methylation in the world of proteins and DNA. It's a small but mighty modification that can have profound effects on gene expression and protein function.
Methylation involves the addition of a methyl group, or CH3, to specific amino acids in proteins or to DNA bases. Don't let its size fool you. This tiny modification can alter a protein's shape, activity, and interactions with other molecules. It can also influence how tightly DNA is wound around histones, affecting gene expression.
Think of it as adding a small, sticky note to a protein or DNA, subtly changing its properties and interactions. While seemingly minor, these changes can have cascading effects on cellular processes.
Methylation is involved in a wide range of cellular processes, including DNA replication, repair, and gene expression. Aberrant methylation patterns are implicated in various diseases, including cancer and neurodevelopmental disorders, highlighting the importance of this tiny modification in maintaining cellular health.
Imagine a sculptor chiseling away at a block of marble, revealing a beautiful statue hidden within. That's proteolytic cleavage in the protein world.
It involves cutting a protein at specific sites, often leading to its activation. This is done by enzymes called proteases.
These molecular scissors recognize specific amino acid sequences and cleave the protein.
Proteolytic cleavage activates many proteins, including those in digestion and blood clotting. For example, proinsulin is converted to insulin through this process.
Imagine tiny bridges connecting different parts of a protein, stabilizing its structure and enhancing its function. That's the role of disulfide bonds in the protein world.
These strong chemical bonds form between cysteine amino acids, creating cross-links within a protein or between different protein chains.
Disulfide bonds are particularly important for proteins secreted from cells or located on cell surfaces, where they provide stability in the harsh extracellular environment. They can also influence a protein's shape and flexibility, affecting its ability to interact with other molecules.
Think of disulfide bonds as molecular staples that hold different parts of a protein together, reinforcing its structure and enabling it to function properly. These bonds are crucial for maintaining the integrity and functionality of many proteins.
Disulfide bonds are essential for the proper folding and function of many proteins, including antibodies, hormones, and enzymes. Disruption of disulfide bonds can lead to protein misfolding and aggregation, which are implicated in various diseases, including Alzheimer's disease and cystic fibrosis, highlighting their importance in maintaining protein integrity.
The symphony of life relies on the harmonious interplay of countless cellular processes. PTMs act as molecular switches, regulating protein function and fate.
Disruptions in PTMs can tip the scales towards disease. Aberrant PTMs are implicated in diseases like cancer and Alzheimer's.
In cancer, dysregulated phosphorylation leads to uncontrolled cell growth.
Understanding PTMs is crucial for developing targeted therapies.